Histology of thyroid and parathyroid glands. Okay. Anyone, have you seen the thyroid gland in dissection hall? In anterior triangle till now you have not seen. Okay. Let's see, tomorrow I may go to the dissection hall. I will show you the thyroid gland. Okay. So first I will give you a brief description. Now, is it complete? I will check. Is it? First, I will give a brief introduction about the thyroid gland, and then we will see the microscopic structure of thyroid gland. Okay. For gross anatomy of thyroid gland, it will be taken separately. Okay. So, which type of gland is this thyroid gland? It is an endocrine gland. Okay. Thyroid gland is an endocrine gland. Okay. Very good. Can anyone tell? Have you studied this in physiology? Thyroid gland? Not yet. Okay. First we will see what, where the thyroid gland is situated, how does it look like, what are the parts of thyroid gland. Okay, and then I will tell you some peculiarities of the thyroid gland. Okay, there are some peculiarities of the thyroid gland. Okay, so first of all, thyroid, the meaning is shield-like. Okay, if you see the thyroid, the meaning is shield-like. Okay, endocrine gland, where it is situated, first of all, it is having rich blood supply and it is situated in the lower part, in front and both the side of the neck. Okay, lower part in front and both the side of the neck. Now, what is the basic function of a thyroid gland? It regulates the metabolic rate or basal metabolic rate, stimulate the somatic and psychic growth of a person. Okay, and very important, it controls the calcium metabolism of our body. Okay, these are some important functions of the thyroid gland. Okay, so basically the metabolic rate, somatic and psychic growth, and important role in calcium metabolism. These are some important functions of the thyroid and parathyroid gland. Now please keep that in mind whenever I am mentioning about the thyroid gland, the parathyroid glands are also mentioned. Okay, along with the thyroid gland, I am mentioning about the parathyroid glands also. Now, this is the only gland. So this is a peculiarity of the thyroid gland is that other endocrine glands are also there in our body. Can you name some endocrine glands? Yeah, pancreas, then which one? The most important one, pituitary, also called as hypophysis cerebri. Okay, from now onwards we'll be calling it as hypophysis cerebri. Okay. Other name of pituitary. So like this, different endocrine glands are there, but among them thyroid is only the gland, endocrine gland, which needs iodine for secretion of the or for the production of the hormones which are secreted from the thyroid gland. Okay, means it is depending on the external source. If it is not getting iodine, it can't produce the hormones. That is a peculiarity of the thyroid gland. Other glands, it don't have such kind of peculiarity. Next, if you see the peculiarity, thyroid is the largest endocrine gland of our body. Okay, thyroid gland jo hota hai, this is the largest endocrine gland of our body. Next peculiarity, this is the only endocrine gland which is placed superficial. Other glands are not superficial, it is deep in the body. But thyroid gland is the endocrine gland which is situated superficial in the neck. Okay, so these are some important peculiarities of the thyroid gland. Okay, other peculiarities are also there, but these are more striking peculiarities. Next, if you see the thyroid gland, it consists of right and left lobes. So two lateral lobes are there joined by a isthmus. Okay, two lobes are there joined by a isthmus, and a third lobe occasionally it is present. Okay, that is called as pyramidal lobe, and which project upward from the isthmus. So from the isthmus it is projecting upward. Occasionally, sometimes it is present, sometimes it is absent. Okay. Next, a fibrous band is attached to the isthmus. Okay, sometimes a fibrous or fibromuscular band descend from the body of the hyoid bone to the isthmus, that is called as levator glanduli thyroidea. Okay, so let me show you the diagram. So here you can see this is the diagram of the thyroid gland. So as I said, the thyroid gland is having two lobes. This is one lobe, this is one lobe. So two lateral lobes, and you can see it is connected by isthmus. So this is one lobe, this is another lobe. Okay, so two lateral lobes connected by isthmus. Okay, next, sometime occasionally a third lobe is present extending from the isthmus, that is called the pyramidal lobe. Okay, If the pyramidal lobe is present, then you can see a fibromuscular band is extending from the hyoid bone to this particular lobe. And what does it call? This is called as levator glanduli thyroidea. Okay. So this is the overall structure. Okay, how it appears, how does it look like? When we will study the gross structure, at that time we have to know about the surfaces, borders of the thyroid gland, along with that the coverings of the thyroid gland, okay, nerve supply, blood supply, lymphatic drainage, everything, as you have studied the other structures. Okay. Now here straightforward I will go to the microscopic structure, that is the histology of the thyroid gland. Now here also you will be noticing one thing, that the structure, whatever we have discussed earlier, if you remember the pancreas, if you remember the salivary gland, the structure almost remains same. That means what is present? Thyroid gland is covered by what? Yes, first of all a connective tissue capsule will be present. So same thing we have studied in case of pancreas also, same thing we have studied in case of salivary glands also. ठीक है, तो यहाँ पे भी same connective tissue capsule is present. Okay, so thyroid gland, first of all it is covered by dense irregular connective tissue capsule, okay, derived from deep cervical fascia or investing layer of deep cervical fascia. Next, parathyroid glands are also present. Okay, so can anyone tell me where the parathyroid glands are present? Yes, अभी किसने बोला? Yes, not behind, actually it is situated on the posterior border. How many parathyroid glands are there? 4. Okay, 2 on each side, superior, inferior, superior, inferior. So see, parathyroid glands are embedded within the capsule on the posterior aspect of the thyroid gland. Okay. Now here also septa extending into the gland in the form of capsule and divided into lobules. So in both the cases we are getting the connective tissue capsule and divided it into lobules. Now if you do a microscopic examination of that particular section of thyroid gland, then what you will see that it is made up of aggregation of follicles. So you have taken a thyroid gland, any lobe, right lateral or left lateral lobe, you have taken a section. If you see under microscope, then what will be visible? It is made up of aggregation of follicles. Okay? Next, what you have to do? You have to see inside the follicle. Now, on microscopic examination, as I said, each lobule is made up of aggregation of follicles. So you have to take one lobule. First will be connective tissue capsule. Connective tissue capsule से septa will go and will divide it into lobules. You will take one lobule. If you see inside, it is made up of follicles. Follicles के अंदर क्या रहता है? Follicular cells are present. Okay. Now follicles, mainly the parenchyma of the gland, consist of spherical masses called thyroid follicles, and size of each follicle is 0.2 to 1 millimeter. That means all the follicles are having not having same size. The size of the follicles are different. So inside the follicle what is present? Follicular cells. ठीक है, follicles के अंदर क्या रहता है? Follicular cells are present. Now it is lined by simple cuboidal to low columnar cells. Okay, simple cuboidal to low columnar cells. Now that depends on the activity of the follicle, whether it is active, whether it is moderately active, or whether it is very active. Depending on them, the size, or you can say the lining epithelium, changes. Next, at the center, you can see a gel-like acidophilic or pink color homogeneous material that is called colloid. Okay, जो follicle होता है, follicle का एक lumen रहेगा. At the center of the follicle, what you can see, a pink color or gel-like acidophilic substance, and that is called as colloid. Now here also the amount of colloid, it varies. Whether the follicle is active, whether the follicle is less active, depending on that, the amount of colloid differs. Next, under H&E staining, the follicular cell shows slightly basal basophilic cytoplasm. Now this is very important because you have to see the follicular cells under microscope. Okay? So when you will see the follicular cells under microscope, especially on H&E stain, at that time what will be the appearance? It shows slightly basal basophilic cytoplasm. Okay? What do you think? What will be the appearance? Basal basophilic cytoplasm. Appearance कैसा होगा? How it will appear? Darkly stained. ठीक है, here I have not used this one. So you have to understand, basal basophilic means it will be darkly stained. ठीक है, तो in the bracket you can write for your convenience. Now single spherical nucleus, and in some cases 1 to 2 nucleoli are present. Next, interfollicular space is also there. What is the meaning of interfollicular space? In between the follicles. Okay, now इसमें क्या रहेगा? In between the follicles, what will be present? Blood vessels will be there, nerves will be there, lymphatics will be there, as simple as that. See, blood vessels, lymphatics, connective tissue fibers, all these are present in between the follicles. Next, a special type of cells are present over there, and these are called as parafollicular cells, or also called as C cells. ठीक है? In between the follicles, the space which is there, that is called intrafollicular space. ठीक है, उसमें mainly क्या रहता है? Connective tissue, blood vessels, nerves, lymphatics. Along with that, a special type of cells are there, and those are called as parafollicular cells or C cells. Okay. So see, they are embedded within a follicle or between the follicles. Now they are polyhedral cell with oval eccentric nuclei, and they are not exposed to the follicular lumen. Okay. Now these are single or small clusters. Mainly you will see the cells in the form of small clusters. Okay. Now these cells are difficult to identify under light microscopy. Now parafollicular cells will show, means when you will see the EM structure, at that time you will be able to notice the secretory vesicles present inside the cells. Now what is the function? The follicular cells secretes T3 and T4, already you know about that, and parafollicular cell secretes calcitonin. Okay, these are the hormones which are secreted by the cells of the thyroid gland. Now follicular cells, as I said, the shape of the follicular cells are not same in all the cases. It differs, okay, depending on the activity of the follicle. Now you can see the follicular cells vary in shape from squamous, cuboidal to columnar, depending on the level of activity of the follicle. Okay, that means highly active follicle is lined by columnar cells. When the follicle is highly active, that means the secretion whenever it is taking place, highly active, at that time it is columnar with scanty colloid. That means cells will be columnar and amount of colloid inside the follicle will be less. Next, when it is moderate active, the follicle is lined by cuboidal and it is lined having moderate colloid. Okay, when it is moderately active, at that time cuboidal and moderate amount of colloid is present. And when it is inactive, that means no secretion is taking place, at that time you can see it is lined by squamous cell and full of colloid is present. Okay, so this is very important. So depending on the activity of the follicle, the shape, or you can say the lining epithelium of the follicle changes, and along with that the amount of colloid which is present, that also changes. Okay, so this is very important, and these things are asked during the time of exam. Now different follicles show different levels of activity. First, follicular cells, already I have told you what is the shape of the follicular cells, basophilic cytoplasm. Now when you will see the electron microscope structure, at that time follicular cells shows presence of apical microvilli, abundant granular ER, prominent supragranular Golgi complex, microtubules, microfilaments. So these are seen under EM structure. Okay, mainly you will see the apical microvilli. Now the apical part of the cell contains many secretory vacuoles also. Let me show you the diagram. So here you can see two diagrams are shown over here, right? So this is the types of thyroid follicle. So which one is the most active? This is the highly active. ये देखो. This is the highly active follicle. This is moderately active follicle, and this is inactive follicle. So can you see the lining epithelium? So see the lining epithelium. Highly active. Also, what will be the lining epithelium in case of highly active? Simple columnar epithelium, and the amount of colloid is less. Next, this is moderately active. So what will be the lining epithelium? Simple cuboidal, and some amount of colloid is present. Okay. Next, if you see the inactive one, simple squamous epithelium, and huge amount of colloid is present. Okay. Next, here you can see the EM structure of the follicular cells. So this is very important. Both the diagrams you have to draw in your practical notebook. Don't forget. Now here you can see the apical microvilli, secretory vesicle at the apical part. Then you can see the Golgi apparatus, lysosomes, okay, nucleus, centrally placed nucleus, and huge amount of rough ER are present. Okay, that is the EM structure of follicular cells. Now, what the activity of the follicular cells change? Just now I have shown you the activity of the follicular cells. Okay, how it will be changing because of the secretion of thyroid stimulating hormone. Okay, that influences the changing of the or activity of the follicles. So from where the TSH secretes? From pituitary gland. Okay. The activity of the follicular cells are influenced by TSH or thyroid stimulating hormone, which are secreted from hypophysis cerebri, or also called as pituitary gland. Now follicular cells secrete T3 and T4. Now T3 means 3 iodine atoms are attached, T4 means 4 iodine atoms are attached. If not, you will be studying in physiology about this, okay, endocrine chapter. Now what are the function of these two hormones? They influence the rate of metabolism of our body. Okay, overall metabolism is influenced by these two hormones, T3 and T4. Okay. Now if you see the function, T3, T4 already I have told you influences the metabolism of our body, and in fetal life and early childhood the thyroid hormones promotes the CNS development. That's why in the beginning I have mentioned psychic growth, or also called as psychosomatic growth of an individual. In intrauterine life and just after birth, the growth and development of the central nervous system is also taking place under the influence of this hormone T3 and T4. Okay? Next, another hormone which is secreted by parafollicular cell. What is the name of that hormone? Calcitonin. Calcitonin. ठीक है, तो इसका फंक्शन क्या है? Lowers the blood calcium level. Okay, तो if you have studied about the parathyroid gland, ठीक है, उसका फंक्शन क्या है? To produce parathormone. Parathormone का फंक्शन क्या है? It will increase the calcium level in the blood, and what it will do, it will decrease, lower the blood calcium level. Okay, so both will act opposite to each other, thus it will maintain the calcium balance of our body. That's why overall you can say that it is maintaining the, or it is controlling the calcium metabolism of our body. So calcitonin lowers the blood calcium level by increasing the calcium deposition in the bones, inhibiting bone reabsorption by osteoclast. So these are the functions, how it lowers down the calcium level in the blood. Okay, this is the main function of calcitonin. Now, this is the schematic diagram of thyroid gland, low magnification and high magnification. So what you can see over here, क्या दिखाई दे रहा है? No. Here in this particular view you can't see the capsule, okay, but you can see the connective tissue septa. Can you see the connective tissue septa? ये देखो. So this is the connective tissue septa, this is the connective tissue septa, right? Now in the connective tissue septa, what is present? Can you see the blood vessels in the connective tissue septa? Okay, I hope you can see these are the blood vessels. See, these are the blood vessels, these are the blood vessels which are present in the connective tissue septa. Okay, along with that other cells of the connective tissue, lymphatics, nerves, all those things will be present in the connective tissue septa. In between the connective tissue septa, can you see the lobules? ये देखो, this is one connective tissue septa, this is one septa. In between these two, this is one lobule, this is one lobule, this is one lobule. Okay, now concentrate into any of the lobule. क्या दिखाई दे रहा है? What you can see in the lobule? Follicles. Okay, तो see, this is connective tissue septa, this is connective tissue septa, this is a lobule. Now inside the lobule, what you can see? Follicles. Okay, you can see the follicles. Now if you take one particular follicle, then what you have to do, you have to zoom it a bit, or that means you have to go under high magnification. Now if you go under high magnification, then this is the diagram. Now what is this, anyone? ये क्या है? This is one follicle. No, this one. This is one follicle. This is one follicle. This is one follicle. Okay. Now follicles are lined by which type of cells? Follicular cells. You can see follicles are lined by follicular cells. Okay. And we have studied about the particular shape of the follicular cells. Okay. It changes depending on the activity of the follicles. In between the follicle, can you see a pink color area? What is this? Colloid. Okay, this is the secretion of the follicular cells. When thyroid hormone is produced, okay, before that the colloid is present inside the follicle. Next, you can see the follicular cells. So these are the follicular cells. Okay. What are the other type of cells present? Parafollicular cells are also called as C cells. So here you can see the parafollicular cells. They are having pale cytoplasm. Now, just now I have mentioned that how you will differentiate or how you will identify which are follicular cell. Follicular cells will be darkly stained. Okay, follicular cells will be darkly stained. Exactly if you say, then what will be the appearance? Basal, basophilic. Okay, basal, basophilic. That means cytoplasm is basophilic in nature. Though it is basophilic, that's why it will be darkly stained. Okay, but if you see the parafollicular cells, they are having pale staining. So like this, easily you can differentiate between the two types of cells. And parafollicular cells kahan pe rehta hai? Where is the situation of the parafollicular cells? In between the follicles. Either they are embedded on the follicles or they are situated in between the follicles. दोनों follicle के बीच में रहता है. As you can see over here, this is one follicle, this is one follicle. In between that, you can see the parafollicular cells. Okay, and intrapericular follicular space which is present. In that space, you can see the blood vessels also. Okay. To see the blood vessels, these are blood vessels which are present in between the follicles. So identification of thyroid gland is very easy. Okay, identification of thyroid gland in the exam, it is very easy, very simple. Okay, till now whatever we have discussed, for example, identification of the tongue, identification of the tonsil, identification of the thyroid gland, those are very easy. Once you know the follicular structure, easily you can identify the cells. Now in this particular case, okay, in this particular case, what do you think, what will be which type of cells will be present over here inside the follicle? अगर colloid ज़्यादा है तो it is inactive. If it is inactive, which type of cell? Simple squamous epithelium. Okay. So this is the drawable diagram which you have to draw in the practical notebook. Okay. Now this is the actual diagram. So this is low magnification and this is under high magnification. What you can see over here? Can you first you have to identify the septa. ठीक है, ये सब जो है first क्या identify करना है? First you have to identify the septa. ठीक है, if capsule is seen then it is well and good. ठीक है, अगर कैप्सूल दिखाई नहीं देता है then what you have to identify? First you have to identify the septa. ठीक है, then see how the septa from where to where it is going. Find another septa, and in between those two septa, see or take one particular lobule. For example, here only you can see, ये देखो यहाँ पे एक septa दिखाई दे रहा है, यहाँ पे एक septa दिखाई दे रहा है. Okay, so this will be one lobule, this will be one lobule. Now inside the lobule, what you have to see? First you have to see the follicles. ठीक है. Next, inside the follicle you will see the follicular cells and colloid. Next, what you have to see? Parafollicular cells. That means in between the follicles, the area which is present, you have to focus on that. Okay. So how the cells are arranged? Parafollicular cells, as I said, they will remain single or cluster. Mostly they are present in cluster. That means cluster of cells will be there. Single cell नहीं रहता है. Most of the cases cluster of cells will be there. ठीक है. और दोनों सेल के बीच में कैसे differentiate करोगे? Follicular cell and parafollicular cell. Follicular cell, the cytoplasm is darkly stained. ठीक है. और जो parafollicular cells है उसका pale stain or you can say bright stain. Okay, like this we can differentiate. ठीक है. Now in order to see or in order to differentiate, you have to go under high magnification. Now this is under low magnification. So here also see here parafollicular cells are seen, but you can't differentiate. Here low magnification, but here it is high magnification. You can differentiate. Okay. So this is how the identification can be done. Now one part which I will not explain you right now, which you will be studying in physiology, that is the production of thyroid hormones. Okay, production of the thyroid hormones. It is there, but this portion you don't have to include in the histology notebook. Okay, this is purely for physiology part. मतलब जो hormones है from thyroid T3 and T4, how they are produced, what are the steps? Because as I have mentioned, there are some peculiarities in the thyroid gland. What is that peculiarity? First one, it is depending on external source for production of the hormones, that is iodine. Okay, without iodine it will not be able to produce the hormones. Okay, so for that the steps are written over here. So this you will study in physiology. Okay, it is, though it is there, that you don't have to write in the practical notebook. Okay, so this is all about the thyroid gland. What is the next one? Parathyroid. As you know everyone that two pairs of parathyroid glands are there. Okay, two pairs means total 4 parathyroid glands are situated. Now where those parathyroid glands are situated? On the posterior aspect of the thyroid gland, very small glands. Now here also connective tissue capsule is there. Okay, each gland has a connective tissue capsule from which septa will extend into the substance of the gland and it will divide it into incomplete lobules. Now within the gland, network of reticular fibers are there, okay, which are supporting the cells, and the parenchyma of the gland is made up of cells arranged in the form of cords. And in relation to the parathyroid gland, you will be able to see that sinusoids are present inside the parathyroid gland. Okay, so parenchyma of the gland is made up of cells that are arranged in the cords, and sinusoids are situated close relationship to the cells. Here you can see the location of the parathyroid glands. So this is the posterior view of the thyroid gland. Okay. So in the thyroid gland, as I have mentioned, how many lobes are there? 2 lobes are there, 2 lateral lobes. So see, this is one lobe, this is one lobe, and it is connected by isthmus. Okay. Now this is the posterior view of the thyroid gland, where you can see the parathyroid glands. So okay, total 4 parathyroid glands are there. Now here also, first of all, each gland is surrounded by connective tissue capsule, same like the thyroid one. Septa will extend and then it will divide it into poorly defined lobules, or you can say incomplete lobules. Glandular parenchyma, the parenchyma part, the secretory part, it is richly supplied by sinusoids. Now these sinusoids are not present in case of thyroid gland. Okay, but ये जो sinusoids होता है, more number of sinusoids are present inside the parathyroid gland. This is a big difference in between these two glands. Now, in case of thyroid also, we have seen 2 types of cells are there, follicular cells and parafollicular cells. Here also, only 2 types of cells are there. What are those? As you can see, chief cells and oxyphil cells. ठीक है, यहाँ पे भी 2 टाइप का cells रहता है. Very simple. So first you have to know about the chief cells or the principal cells. Now the parathormone which is secreted by the gland, now that is the parathormone. Okay, now this chief cell secrete parathormone, or also called as parathyroid hormone. The short form is PTH. Now these are more in number than the oxyphil cells, so mostly you will be getting the chief cells. Under light microscopy or compound microscope, the chief cells are polygonal shape with central located nucleus, dark staining cytoplasm. Here chief cells, dark staining cytoplasm, very easy to identify. Now this dark staining cytoplasm is depending on the amount of glycogen and lipofuscin, and based on that it is divided into light, dark and clear cells. Okay. तो जो chief cells होता है also called as principal cells, more in number. What is the main function? To secrete parathyroid hormone or parathormone. ठीक है? What is the identifying feature? Darkly stained cytoplasm. Dark stained cytoplasm. Okay. Now depending on the amount of glycogen and lipofuscin, okay, which is a yellow color pigment. The cells are divided into three types. What are these? Light, dark and clear cells. Under EM structure, you can see abundant rough ER, Golgi apparatus, secretory vesicles are present inside the chief cells. तो chief cell का function क्या है? तो it will secrete the parathyroid hormone, or also called as parathormone. Now parathormone का function क्या है? It will increase the serum calcium level. How it will increase the serum calcium level? There are some mechanism. Okay, as you can see, increasing the bone reabsorption through stimulation of osteoclastic activity. Okay, so I hope you have studied about the bones of cells of the bone. Osteoclast का function क्या है? Removal of the bone. Okay, so it will enhance or it will increase the osteoclastic activity of the cells. As a result, what will happen? More amount of bone will be released. Now calcium reabsorption from renal tubules and calcium absorption from the gut. So whatever we are eating, in that also some amount of calcium is there. For example, milk is a rich source of calcium. So whenever a person is taking milk, from the gut, the absorption, when it is taking place, this particular parathormone, parathyroid hormone will increase the rate of absorption for calcium. Okay, so this is some mechanism by which it will increase the serum calcium level. And which will decrease the serum calcium level? Calcitonin. Calcitonin is secreted by parafollicular cells of the thyroid gland. Okay, like this the balance is maintained. Next, oxyphil cells. So can anyone tell me what will be the function of oxyphil cell? कोई function नहीं है. Okay, it is there. Okay, but it may have some function, but till now the function is unknown of the oxyphil cell. Research is going on. So oxyphil cells, these are less in number but much larger in size. So the size of the cell is larger. Okay, either they appear single or in the group. Light microscopy, if you see under light microscope, these cells have abundant deeply eosinophilic or acidophilic cytoplasm. Okay, now what is the meaning of acidophilic cytoplasm? That means ये जो particular cells है, it will under H&E stain, it will appear pink in nature. Pink color का appearance होगा. Okay. See, oxyphil cells have deeply eosinophilic or also acidophilic cytoplasm because of large number of mitochondria. So that is the appearance by which you can identify. And the size of the cells are larger. Okay, oxyphil cells का जो size है वो larger होता है as compared to the principal cells or chief cells. Now secretory vesicles are present. Okay, very less rough ER are there. Okay, they don't have any secretory function, and the function of the oxyphil cells are unknown. Still, it is unknown. Now secretory vesicles are there, but why it is there? इससे क्या secrete होता है? Till now, it is unknown. So this is the microscopic structure. This is the schematic diagram, and this is the actual diagram of the parathyroid gland. This one you have to draw in your practical notebook. ठीक है, तो what you can see over here, starting से देखो, first you have to see about the capsule, connective tissue capsule. तो capsule यहाँ पे दिखाई नहीं दिया, capsule is there. See, connective tissue capsule is there. After that, can you see the septa? ये देखो, this is the septa, this is septa. Okay, now here it will divide into lobules. So see, this is one lobule, this is one lobule. So these are numerous lobules. ठीक है, inside the lobule what you can see here, follicles are not seen. Instead of that, you can see mainly two types of cells are there. What are these two types of cells? Chief cells and oxyphil cells. Now the question is, how you will identify chief cell and how you will identify the oxyphil cells? Depending on the staining properties easily. Function तो है, but identification depending on the staining properties. Can you see the see, these are the chief cells. So first you have to notice one thing, number of chief cells are more, number of oxyphil cells are less. First point. Okay, I think it will be better if you note it down. Number of chief cells are more. Number of chief cells are more, number of oxyphil cells are less. First. Second point, if you see the size of the chief cells are smaller, size of the oxyphil cells are larger. Okay, first is the number, second is the size. साइज किसका ज़्यादा है? ऑक्सीफिल सेल्स. चीफ सेल का साइज छोटा होता है. ओके, नेक्स्ट, कैन यू सी द स्टेनिंग डिफरेंस? एवरीवन प्लीज फोकस ओवर हियर. ये देखो, दिस आर द चीफ सेल्स, दिस आर द ऑक्सीफिल सेल. तो स्टेनिंग डिफरेंस क्या होता है? Yeah, you can say darkly stained, brightly stained, but here you can see here the cytoplasm is acidophilic, or also you can say eosinophilic. Okay, but here it is basophilic in nature. So like this you can easily identify. Okay, so this is the points which you have to notice. Next, in the connective tissue septa. Okay, next in the connective tissue capsule, first of all, blood vessels, you can see these are the blood vessels, these are the blood vessels. Okay, though it is a endocrine gland, that's why we'll be able to see huge amount of blood supply, blood vessels. Okay, so see, these are the blood vessels. Now in this case, these are blood vessels, these are blood vessels. Okay, but here mostly we are getting sinusoids, as I have mentioned earlier. ठीक है, तो ये जो dilated parts है, this dilated parts, this will be the sinusoids. Okay, so here also you can see blood vessels are written, but you can say that these are sinusoids only. Okay, ये देखो. Okay, next, this is the original diagram, or you can say low power, low magnification image of parathyroid gland. Now here, how will you identify the auxiliary cells and how will you identify the chief cell? See the appearance of the chief cell and see the appearance of the auxiliary cell. Okay, staining difference is there. Clearly you can see the chief cells and you can see the auxiliary cells. And in the auxiliary cells, ये जो lipofuscin एक येलो कलर का सब्सटेंस होता है. ओके, एज द एज इंक्रीजेस, द अमाउंट ऑफ दैट पर्टिकुलर लाइपोफ्यूजन इंक्रीजेस. इट डिनोट्स दैट द सेल्स आर गेटिंग ओल्डर. ओके, okay, so that is all about the parathyroid gland. Okay, so we'll go to the lab and we'll see the slides of thyroid gland.